So, in today's session, first of all, let me give my brief introduction. I am Dipanshi Avasthi and I am a PMRA fellow. I'll be your TA for this course. So, in this session, today we'll be going to discuss about the week one content and practice few MCQs related to that. And if time will allow, then we will also try to look for the week two concepts as well. So in week one, you have covered the fundamentals of the central dogma in which you have studied about DNA, RNA, proteins, mutations. You have also studied about the chromosome structure, its functions, organization, structure function relationships, chromosomal abnormalities. So here in the bottom, I have mentioned the reference book, which sir has already mentioned you. So I am taking the content from there. So in this session, we'll be just revising all these things which you have covered in week one, and then we'll be working on the MCQs. So wherever you will be having any doubt, you can ask me in between. And if my content is not visible or if something happens, then you can inform me so that I could know. Okay, so let's begin with the central dogma. So what is central dogma? It is generally the flow of information from DNA to RNA and further RNA to protein. So you can look it in the form of genotype to phenotype conversion. So genotype means the DNA content which you have with yourself, and then it converts to RNA and further it forms a protein which shows a visible expression in a human. So that is a phenotype which you observe. Genotype you cannot observe, but the phenotype you can observe, which is as a result of a genotype. So this is known as central dogma, which means flow of information from genotype to the phenotype. So normally the DNA is present in the nucleus if we talk about the eukaryotes, and this DNA with the process of replication it divides into its own copies. So DNA to DNA conversion is known as replication, with which it maintains its number. Whenever cell division occurs, then DNA make its identical copies. Similarly, the transcription, which is the formation of RNA from the DNA, it also occurs inside the nucleus where the single strand of DNA, which is the antisense strand, or you can say the template strand, converts to RNA. So this is transcription. And further, this RNA contains many coding as well as non-coding fragments. So various processes occur such as splicing. And after splicing, the process of translation, which is the formation of protein from the RNA, it occurs in the cytoplasm. So, because all other proteins such as ribosomes, which are essential for the synthesis of protein, they lie in the cytoplasm. So that's why this RNA moves from the nucleus to the cytoplasm for the synthesis of proteins. And when it moves to cytoplasm, then there are certain things. So RNA cannot move directly after formation to the cytoplasm. Before that, it undergoes splicing as well as few processing such as tailing as well as capping. So after that, it moves to the cytoplasm where different ribosomes, tRNAs, different proteins comes, and that allow that RNA to translate into a amino acid stretch which forms a protein. So in this way, the information is being sent from the nucleus to the cytoplasm, and then it's affecting the entire machinery. So this is all about the central dogma. So let's see its first question, which is related to this. So the question is, the central dogma of molecular biology is an explanation of the flow of genetic information within a biological system based on your knowledge of the processes involved and the cellular compartment where the process takes place. Choose the option that identifies the correct pair. So in this, you have to read these options and tell which option is the correct one. So first option is replication. And then the next thing suggests the place where it occurs, so in nucleus, and the next thing represents what happens in the process. So that is copying of DNA into RNA. The second option is transcription occurring in cytoplasm and copying of DNA into RNA. The third one is transcription occurring in nucleus, copying of RNA into protein. And the last option is translation, which occurs in cytoplasm and it copies the RNA into protein. So, as we have discussed about central dogma, so what do you think? What would be the probable answer for this question? So now you
And
can, which one? Fourth?
fourth,
Okay, somebody else wants to tell the answer? Wait, yeah. Anyone else? wants to give answer to this question?
Madam, even A is correct. A and D.
A. So is my screen still visible, right? The
Yes,
presentation one. Okay, so you're saying A
madam,
option.
madam, what is meaning of dogma? I, is a English word or something else? It is taken. Dogma means what? I think
Actually,
I didn't heard anywhere in English literature. This is only we hear in molecular biology.
yeah, I guess I'm not sure about its origin, but maybe it's a Latin word. I'm not sure from which language it is derived, but it means the flow of information. Means dogma is a collection, the main major collection, or you can say the flow of information of. So that's why it's called central dogma.
Because it is clearly saying it is a flow of information from genetic to biological system.
Yeah,
But based on your knowledge, it is asking about you us about cellular components. So unless genetic material will be transferred from DNA to RNA,
yeah,
translation will not occur.
yeah,
Actually,
right.
question either question is wrong or option is wrong. Actually, it should have been replication. It is
Okay.
occurring in nucleus and copying from DNA to RNA. Because since all 3 days everybody is saying D, but whenever it is a genetic information, it should come from DNA.
Okay, so actually, why A option is not correct? Because replication is the process in which DNA is converted to DNA itself. So A option cannot be correct because it mentions that
Okay.
copying of DNA into RNA. So this process has
Okay.
another name, that is transcription, which means conversion of DNA into RNA. So A option would have been correct if it would have been written copying of DNA to DNA. So we cannot say that A option is correct. Then if we look at second option, that is transcription, and it says that it happens in cytoplasm. So from here only it's wrong because transcription occurs inside the nucleus. So we can reject this option. Then if we look at the third option, which says that transcription, nucleus, and copying of RNA into protein. So transcription is the conversion of DNA to RNA, not RNA to protein. So third option cannot be correct. Then if we look at the fourth option, that is translation, it occurs in the cytoplasm and copying of RNA into proteins. So this is the correct one because this RNA into protein happens in the cytoplasm. So that's why option D will be the correct answer.
Madam, whenever we have to identify correct pair means two things. Actually, here there are triplets. No? Translation, cytoplasm, as well as copying of DNA.
Yeah, here three things you have to keep in mind. You cannot select on the basis of by looking at the 2 points only. So
Pair means 2 points. No? Okay.
okay, so you mean that identifies the correct pair I've mentioned. So that's why you're saying
Correct
okay.
triplet.
Yeah, sure. You know, I haven't edited the question, I just pasted it. So, okay, let's again start with the next slide. So, I hope this question is clear to you that why the answer will be D. So, if there is no further doubt, then I'll proceed to the next slide. So, when we talk about the cell, so we say DNA. So, in nucleus, you don't see DNA clearly. DNA is very long, so it is not present as a form of thread. It's compactly arranged in the form of chromosomes. So, if you focus on the nucleus, then you will see that there are chromosomes which are present inside the nucleus, and these chromosomes are nothing but these are the DNA structures which are compactly packed. In this, the DNA is wrapped around the proteins which are named as histone proteins. So, when the DNA is wrapped around the histone proteins, it is an octameric protein and it forms a structure called nucleosomes. So when you further untangle this chromosome, then you will observe the other structures in which the DNA is wrapped around the histone protein that is known as nucleosomes. And when you further remove this DNA segment from the nucleosome, then you will observe there will be genes. And further, these, if we look more closer to it, then you will see the nucleotides. So nucleotides are nothing but the building blocks of DNA, which means sugar, phosphate, and the nitrogenous base, which constitutes the DNA, which are arranged in a certain manner which forms the DNA. So these are the nucleotide, and it is called nucleoside if phosphate group is not present, then that is known as nucleoside. So I hope you know about the nucleotide and nucleoside concept. And then when we talk about DNA, then we mention four nitrogenous bases that are guanine, cytosine, adenine, and thymine. So now let's focus more over the DNA, that how it is arranged and what are the things that are present, because it is the first part of our central dogma. So DNA stands for deoxyribonucleic acid, and how it got this name, because there is sugar which is deoxyribose sugar which is present in this. That's why it got its name as deoxyribonucleic acid. It is localized in the nucleus in eukaryotes, but when we talk about prokaryotes, then they do not have nucleus. So in them, the chromosome is arranged in the cytoplasm. That is not in the nucleus. And other than this, if we talk about eukaryotes, then you can find DNA inside the mitochondria as well as chloroplast as well. And the backbone of the DNA is made up of sugar and phosphate polymer, but it is the sequence of bases attached to these sugar molecules which determines the identity and genetic function of any DNA sequence. For example, in all the organisms, the DNA is made up of these building blocks. But what is that component which distinguishes one person from the another is the bases, that which bases are arranged. So in this way, it gives uniqueness to a person, this nitrogenous bases. Now, DNA normally occurs as a double helix, and it comprises of the two strands which runs anti-parallel to one another. Anti-parallel means if one strand is from 5 prime to 3 prime, then another strand will run from 3 prime to 5 prime direction. And then these two strands are joined together by the hydrogen bonds. So this hydrogen bond pairing is involved. And now if we talk about the nitrogenous bases, then there are two types of nitrogenous bases. One are purines and other one are pyrimidines. If we talk about purines, then these are the double ring structures and pyrimidines are the single ring structure. In purines, the amino acids which comes are the adenine and guanine, whereas in pyrimidines, the nitrogenous bases that come are cytosine and thymine. So adenine always pair with thymine with the double bond, if you can see here, and cytosine will pair with guanine with the triple bond. So let's see that. So here I'm showing you the bonding present between adenine and thymine. So it is a double bond and it is a hydrogen bond. So you must remember that whenever adenine and thymine join to one another, then there are two hydrogen bonds formed. And when the guanine joins to cytosine, then there will be formation of three hydrogen bonds. So why we are discussing these hydrogen bonds? This is because a more number of hydrogen bonds will be present between the two nitrogenous bases. It will make that bond very strong and difficult to break. So, if we compare the DNA of ancestors, or you can say in the olden time, when during the evolution, you will see that the extremophiles, which used to survive in the extreme environments, so they have greater number of GC content as compared to the means present day organisms. So these primitive organisms have that higher GC content. So this is because this GC content has the triple hydrogen bonds, which are strong bonds and which do not break easily. So there is a process which is named as denaturation. So denaturation means as the two DNA strands are connected to one another with the hydrogen bonds, so at high temperature, these hydrogen bonds break and the two strands denatures and separate into two single strands. So that is denaturation. So there is a question that which of the two given duplexes do you think will denature at a higher temperature? So there are two duplexes given. So observe them carefully, observe their nitrogenous bases, and can you answer that which of the given two duplex will denature at higher temperature? And this is related to this hydrogen bond concept. So if you
The
can
second
answer
one. The
Second
second one.
one.
second one, okay. So yeah. So why second one? Uh, Actually, it's already
Due to the
visible.
presence of higher triple bond.
Yeah. So wait, let me admit all others. Yeah. So why is the second option? Somebody was answering.
Due to the presence of more triple bond in it.
Yeah. More number of means higher number of hydrogen bonds. Triple hydrogen bonds will be present in GC and greater number of GC are present. So that's why.
Ma'am,
Yeah.
ma'am, could you please say again that GC content and the course of evolution you have said just
Yeah.
now?
Yeah. 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 I'll repeat. So I'm saying that the primitive organisms which used to live in the extreme environment, so when we compare the DNA from the primitive organism and the current organisms, for example, if we talk about the bacteria, so archaebacteria and the current bacteria or current eukaryotes, then we will observe that in their DNA there is higher GC content, because this GC content provides the stability to the DNA at the higher temperatures, because more number of CG will be present, higher number of triple bonds will be present, and those DNA will be difficult to denature, because if DNA will denature, then this whole cellular machinery will collapse. So to avoid the denaturation, in the primitive organisms there were greater CG content. So as per studies, it is mentioned that this CG content provides stability to the DNA at the higher temperatures. So that was I was mentioning. So is it clear?
Madam, there are 18 bonds in duplex A and 22 bonds in duplex B. So what happens if the same temperature is applied to both? So 80 content of both will go, or only GC content will remain?
Sorry, 80 content what? Actually, these
This
two strands are not
structure.
like, means these two strands are joined together. So if you are going to separate, denaturation means complete separation. So if means it doesn't mean that it will separate from the middle. Are you getting it? It means that if multiple CG are present, then it will let the strands fixed, even if we provide high temperature to it, but still it will not denature. Even if the ATs will denature a little bit, but still that CG is holding it tight, so they will again renature. So it is the ability of DNA that it renatures back. So even if certain ATs are present in this second duplex, but still, if we provide the same temperature, the ATs will try to denature, but that CG content, it will let it hold to the fixed position. So it will not let it get separate. So in that way, this even if we provide the same temperature, the second duplex will not denature as easily as compared to duplex one. So I hope.
Because it is because of formula that A always pairs with T and G always pairs with the C.
Yeah, because there are donor groups present, right? Hydrogen bond donors and hydrogen bond acceptors, due to which they form bonds. But in certain cases, due to chemical
Because one of them
agents,
is
yeah,
purine and one of them is pyridine.
pyrimidine, yeah, right.
So, purine can pair with pyrimidine. So A cannot pair with C and G cannot pair with T. So is there
Yeah.
any that molecular formula is there or some other thing?
Yeah, this thing happens because of the interactions. So in A, there are only, if you see here, one is hydrogen bond donor and one is hydrogen bond acceptor in adenine. If you can see in this figure towards the left, in adenine you see. So because in adenine can't pair with cytosine, because in that there are three groups. One is hydrogen bond acceptor, then one is nitrogen, which is making sharing this hydrogen with the guanine molecule. So this happens due to the interactions or the interacting partners which are present on the particular nitrogenous base. So adenine can give one hydrogen and accept one hydrogen, due to which it pairs with the thymine and not with cytosine. Because otherwise, there will be the conflict in the structure. Means because interactions will be present, now there will be repulsions, steric clashes. So due to that, due to the chemical interactions, you can say uh, the adenine is more stably bounded with the thymine as compared to cytosine. Hence, it is favored. But yeah, with
Uracil is also pyrimidine.
yeah, uracil is also pyrimidine, but that is present in RNA, so we are not discussing it here. It is exactly same as thymine, except for this methyl group that is absent in the uracil, which is present in thymine. So that is the single difference that is present in thymine and uracil. So in RNA, uracil will pair with adenine and not the thymine. So is it clear?
Yes, madam. Clear. Thank you.
So if there are no further doubts, then let's move to the another slide. So now we are talking about DNA, then we also hear about genes. So what are genes and where are they present? Because now we are talking about DNA. So the genes are nothing but they are the discrete segments of DNA. So that means if the DNA is very long, then there could be multiple genes that can be present on that DNA. And genes are the discrete segments of the DNA which are used as the template to synthesize a functional complementary RNA molecule. So these are the genes which converts to the RNA molecule. Not means it is not that the entire DNA is going to convert to RNA, but there are certain chunks of the DNA which are referred to as genes, which converts to RNAs and further form proteins. And it's not absolute that the RNA will form the proteins. There are certain genes which codes for the RNAs which do not code for the proteins further. So you can say the known protein coding genes. So there are certain RNAs which have some regulatory roles. So that are the things. And yeah.
genes will always have a sensible part, or will always have a message, or any triplet we can call it as a gene.
So if we talk about the genes, so genes, it will not always code for a protein. So if we talk about if you are saying sensible part, so what people mean with the sensible part, that every gene will code for some protein. So that is not the case. In certain cases, these genes have some regulatory role. So they will form some RNA, which, for example, if you talk about rRNA or tRNA, because it is the messenger RNA which carries the message and which will form the protein. But there are certain other RNAs as well, such as transfer RNAs or the ribosomal RNAs and many other RNAs as well, which do not code for protein, but they have some regulatory role. So means genes will code for those RNAs as well. So if we say genes, so it means that the part which can code for the RNA. So is it clear?
Thanks.
Yeah. So uh, okay. So as I've already discussed that all the genes will not code for the protein coding RNA. Certain RNAs will be having regulatory role, but we will still call those as genes, even if they are not coding for the protein. So here's another question. I think it was mentioned in the assignment 0 if you have attended that assignment. So the question says, which one of the following statements accurately explain the relationship between the parts of genetic materials discussed? So now we have to read these statements carefully and check which one is the accurate statement. So first is each DNA molecule contains many genes. Next is each DNA molecule contains many chromosomes. Third is each chromosome contains many DNA. And the fourth one is each gene contains many genes. So this thing I've already discussed. So what do you think? What will be the probable answer to this question?
Option A.
Option A. Anyone else wants to answer?
Yes, ma'am. A.
Okay. Okay. Anyone else? Okay, fine. So, yeah, A will be the correct answer because each DNA molecule contain many genes. And why B can't be correct? Because they are saying each DNA molecule contains many chromosomes. But we have already seen that chromosome is nothing but it is the compact packing of the DNA. So this can't be true. Then there is third option that each chromosome contains many DNA. So this is also not true because the chromosome is made up of only single DNA. And then there is each gene contain many genes. So this is absolutely wrong. How each gene can contain many genes. So hence the right answer will be the option A. So let me just check. Okay. So now let's move to the next slide. So there is another question. So if we talk about genes, then we'll also talk about alleles. So do you have any idea about the allele? What is allele? Because in genetics we talk very frequently about the allele thing. So let's first discuss this question, then I'll ask you. So an allele is option A mutant version of a gene. Option B, a variant of a gene that may not cause a disease. Option C, one of the several variants of a gene present in normal population. And option D, all of these. So now tell me, have you heard about the name before, allele?
Yes, ma'am.
Okay, so what do you understand by allele? Means if you
Ma'am,
have, yeah,
alternate form of a gene.
right, true. So now look at this question and what do you think would be the right answer?
Madam,
Yeah,
in simple thing, it is one of the partner of a pair. If there are two is maybe
right.
pair, maybe one may be strong, another may be weak, but one of the two is called as allele. It is a one
Yeah,
partner
so
of pair.
yeah, in simple words, we
Very
can
simple.
say allele as that. So if we talk about the traditional definition, then it is the alternate form of a gene. And but what you mentioned is also true. In simple words, we can say that one of the pair among the gene. So it is known as allele. So now let's look at the options. So it is saying a mutant version of a gene. So if we call allele, so it can be a mutant version of a gene because a gene have a pair. A gene exists in a form of a pair, and each of its pair is referred to as allele. So one pair can vary from, one you can say one allele can vary from the other. So it is a mutant version of a gene. Means both the alleles vary from one another. They can be mutant. One allele can be mutant of another allele. So in this way, a mutant version of a gene could be correct. So next is a variant of a gene that may not cause a disease. So it is also true because when we talk about diseases, then we see that there are two alleles. If we talk about, for example, dominant and recessive thing, so we say that if a disease lie on the recessive allele, then it will not cause a disease until and unless both of the alleles are recessive in nature. So in that way, here they have mentioned a variant of a gene that may not cause a disease. So yeah, allele can be a variant of a gene which may not cause a disease. So it is also true. Then third is one of the several variants of a gene present in normal population. So yeah, it is also true because it could be the one of the several variants of a gene because there are certain genes which can have more than one variant. So yeah, it is a single one of a several variant of a gene. So it is also true. So hence the correct option will be the option D, which means all of these. And it is also self-explanatory as I mentioned that what is allele. So here to more elaborate this thing. So first of all, this is the chromosome. These are the two homologous chromosomes which are you can say which are almost identical to one another. But there are certain alleles or the genes means there are two form of genes which are present in either of the homologous chromosome. So if you see here small d and capital D, so these are the alleles. And the gene, if we say gene, then gene is gene D, but small d and capital D are two alternate form of the same gene, which is D. So in this way, this is a allele, and this is entire is a chromosome, and the pair of a chromosome is referred to as homologous chromosome, which have means identical genes present on it. And then in the middle, you can see it's a centromere, which joins the two chromatids of a chromosome. So chromatids are the two shorter fragments which are joined to one another at the centromere. And in this, here also you can see there are multiple genes present in it. So gene D, E, F, G, H, these are genes. And in each homologous chromosome, you see two different form of genes, which means it is a allele. So capital D and small d is a allele of a gene D. So I hope now it is clear that what is gene, what is chromosome, what is allele. Do you have any doubt about allele, or should we proceed
No,
further?
ma'am.
No,
Okay,
ma'am.
okay, fine. So now let's look at another question. So this is related to the chromosome, about the chromosome structure. So it says the region of the chromosome that joins a pair of sister chromatids is known as dash, and the chromosome ends are referred to as the dash. So now the options are centromere, promoter. Option B is telomere, spindle fiber. Option C is spindle fiber, centromere. And option D is centromere, telomere. So in first you have to tell that the two sister chromatids are joined by a particular region. So what is that region called? So in first you have to tell about it.
Centromere.
Okay, right. And in next part you have to say that the ends of the chromosomes are referred to as.
Telomere.
Telomere.
Yeah, very good. Right. So the right answer will be option D, which is centromere and telomere. So as we have observed the structure earlier, so in that we saw that the centromeres join the two sister chromatids and the terminal regions or we can say which are present at the terminal positions. These are known as telomeres. So option D will be correct. So now let's move to the RNA, which is the next component of the central dogma. It stands for ribonucleic acid. And so when we talk about RNA, it is also a genetic material in certain viruses. So when primitive organisms were studied, so earlier there was a big debate to understand that what is the genetic material. Different scientists performed experiments because in primitive organisms it was found that RNA could be a probable genetic material. So how we landed on to calling DNA as a genetic material and not RNA. So when different experiments were performed, then it was found that DNA is more stable than RNA, and hence DNA is the genetic material. And further, DNA can transcribe to RNA. So DNA and RNA are almost similar. There are few differences which exist. First is that DNA is a double-stranded and RNA is single-stranded. Then the difference exists at the level of sugars. So in DNA, the sugar which is present is deoxyribose sugar, whereas in RNA, the sugar which is present is ribose sugar. So it is a 5-carbon membered ring. So this second position, second carbon of this sugar determines that either it is deoxyribose or ribose. So in ribose sugar, the OH group will be present at the second carbon of this sugar, whereas in deoxyribose, you will see that the OH group is missing. Only a hydrogen is present. So that's why it is known as deoxy, means oxygen is absent at the second position. So this is the next difference. And the third difference is at the level of nitrogenous bases. So adenine, guanine, and cytosine are present in both the DNA and RNA, but the base which varies is the uracil and thiamine. So thiamine will only be observed in the DNA, whereas uracil will be observed in the RNA. So this is the main difference between the RNA and DNA. Now we'll look at the process of transcription, which means conversion of DNA to the RNA. So when this process occurs, then note means if we see, then both strands of DNA will not participate in transcription. There is only a single strand that participates in this process and which forms RNA from the DNA. And that strand is referred to as template strand, or you can say antisense strand. So firstly, I'll tell you that why it is called template. So if we say template, template means that something is acting as a base for the another thing. So template means because it is acting as a template, and by looking at it, its complementary RNA will be synthesized. So hence it is known as template strand. Now it is written antisense. So many people get confused that why it is antisense, because it is coding, then it should be a sense strand. Why not means sense? Why it is antisense? So the reason behind this is that the RNA which will be formed from this template strand is exactly similar to the sense strand, except for the thiamine and uracil, because it is complementary to the template strand. So sense strand is also complementary to this template strand. So hence the RNA which is formed is exactly same as the sense strand. And the difference will only occur at the thiamine and uracil. So those will be replaced in the RNA. So hence the strand complementary to template is referred to as sense strand, and the template strand is referred to as antisense. So that is the reason that why these names were given. So now RNA will be formed from the strand which runs from 3' to 5' direction, because transcription, replication, as well as translation, every process occurs from 5' to 3' direction. So whenever a new thing is being synthesized, that will be synthesized from 5' to 3'. So in this way, the DNA which will be involved, that will be running from 3' to 5', and the RNA that is getting synthesized, that will be synthesized from 5' to 3' direction. So always keep this thing in mind. Now there are different kinds of RNAs. As I was mentioning earlier about the genes, that genes are coding for the RNAs. So not all RNA will be protein coding. So there are different types of RNA. First is mRNA, that is messenger RNA. So it got its named as messenger RNA because it is carrying a message. So it codes for protein. Then comes tRNA, which is transfer RNA. So why transfer? Because it is involved in the protein synthesis where it is transferring the amino acids. Because protein is nothing but a chain of amino acids joined to one another by the peptide bonds. So hence tRNA is involved in the translation process in which it transfers the amino acids. Hence it got its name as transfer RNA. Then the third RNA is the ribosomal RNA or rRNA. It got its name because it is present in the ribosomes. So hence it got its name as rRNA. Then there are certain other RNAs which are snRNAs, small nuclear RNAs, snow RNAs, which are not present in the major chunk. These are very few in numbers because they are occurring inside the nucleus or the nucleolus. So similarly, the snow RNA, which is small nucleolar RNA, that is present in the nucleolus. Then comes miRNAs, that are microRNAs, which are very small, and they have role in the regulation process. So these are generally regulating the gene expression. Either they can silence the gene expression or they can allow it to express. Then there are small interfering RNAs, that is siRNAs. They are also involved in silencing the gene expression because whenever they'll be binding to the RNA, then RNA will not be able to translate. So hence, in that way, they are just silencing the gene expression. Then there are certain long non-coding RNAs which are not coding for anything, but they are regulating the gene expression. So in this way, there are different kind of RNAs which are produced inside the cell, other than mRNA, which is going to produce the protein. So now we'll be focusing majorly on the mRNA because it is coding for the protein. So let's see this question. A newly synthesized RNA has the following sequences, that is 5 prime, AUC, CUG, GUA, 3 prime. Which one of the following options identifies the correct sequence of the transcribed DNA template? So now you have to tell the sequence of the DNA strand. So the DNA template strand. So keep this thing in mind that a DNA template which is synthesizing this particular RNA sequence, what would be that DNA strand? So the options
Option
are
A.
option A, why?
Because it will be converted from 5 dash to 3 dash, and uracil will be converted into thiamine. No, thiamine
My
will be
option will
converted
be
into
B.
Okay, somebody is saying option B. So any other guesses?
Yes, ma'am. Option B.
Option
B
B.
is correct.
So first of all, they are mentioning,
Option C.
okay, option C, fine. Uh, so now let's see what will be the correct answer. So in this, first of all, let's read again. Let's again read this question to means get clarification. So in this, they are saying that which one of the following option identifies the correct sequence of the transcribed DNA template. So in this, they are asking you about the sequence of the transcribed DNA template. Okay, so it will be the antisense strand, right? And antisense strand always run from 3 prime to 5 prime direction. But in the options, they have given you 5 prime to 3 prime direction DNA templates. So now to see it, if you see, this is the RNA. So what would be the complementary DNA strand? That would be TAG, GAC, CAT. But if we see, then these strands they have given in the options, they are from 5 prime to 3 prime. So now we have to convert it from 5 prime to 3 prime. So we'll observe that option B will be correct because it is the complementary to the RNA being synthesized in the 5 prime to 3 prime position. So if anybody have doubt, then they can ask. Is it clear that why we option B is correct? If anyone have doubt, then I can again explain. And if there is no doubt, then we can proceed further. Is there any doubt?
I think no, ma'am. Please
Okay,
proceed.
okay, okay. Yeah, so let's proceed with the next. So now this transcription thing is also clear. So now we'll be moving to the process of splicing and why it is important because now we'll be talking about translation after after RNA it comes proteins. So before moving to proteins, what happens that the RNA is getting synthesized in the nucleus, so it has to move to the cytoplasm. So before that, there is a process called splicing in which the non-coding regions which are present in the RNA they are spliced out or removed. So if we talk about the RNA, so this is the transcription unit which you can see in the A. So it represents the gene. So gene which is coding for the RNA. So if its transcription occurs, then the B is synthesized, that is the RNA molecule. And this RNA is known as precursor RNA or pre-mRNA, you can say, not only mRNA, but it could be any RNA. So precursor RNA or heterogeneous nuclear RNA is also a term which is named, where the RNA is not processed. It contains introns as well as exons. So exons are the coding regions and introns are the non-coding regions that are present in the RNA, and we need to remove these known coding regions. So this process of removing the known coding region is known as splicing. As the name is clear from the word the splicing. Splice means to cut. So splicing means to cut the intronic regions and then joining back these exonic regions, and hence the mature RNA will be synthesized, and now it is ready to get transported to the cytoplasm. But before that, there are certain processing which will be undergoing in the nucleus. That will be the tailing and the capping. Because tailing and capping are important because they are going to protect these RNAs from the attack of other enzymes, such as ribonucleases, which cleaves these RNAs or the DNA molecules. So to protect it from this enzymatic damage, its capping and tailing is important. And after that, it gives signal to the machinery that now it is ready for the transportation from nucleus to the cytoplasm, and further it can code for the protein if it is mRNA. So there are two mechanisms of splicing. First is the intron-based self-splicing, in which the introns automatically themselves get cleaved out. So these introns have three parts. One is the 5' splice site, then the branch point, and then the 3' splice site. So 5' splice site is the region that lies on the border of exon and intron. So the 5' position of the intron will contain the sequence that is GU. It is acting as a signal sequence. So whenever the means in this case there is an intron based self splicing, so no other agent is coming to identify that, but otherwise this is the splice donor site that is recognized by the agents that are causing the splicing other than this cell splicing process. So in this there is a nucleophilic attack, and hence by this machinery the introns are getting out spliced out by themselves without the use of any further external proteins. So this is one mechanism of splicing. And then the another mechanism is the snRNP based mechanism, in which the small nuclear ribonucleoproteins, that is NERPs, so they are also referred to as NERPs. So they are involved in the splicing, in which they comes and bind to these regions. So there are five type of these spliceosomes. So this is also known as spliceosomal machinery. So snRNPs are the small nuclear ribonucleoproteins which constitutes the machinery which is known as spliceosomal machinery. So why spliceosome? Zome is termed when there is a complex, and splice because it is involved in splicing. Hence, all the nerves that are involved in the splicing process are termed as spliceosomes. So this is a spliceosomal machinery which is acting and which is going to remove these introns from these exonic chunks, or you can say from this precursor mRNA. So in this way, U1, U2, U4, U5, and U6, U3 is not present. So these all together, they bind to the introns, they bring together the introns, and then they cut off the intron from the exons. And at last, these exonic fragments will bind together, and then it will be a mature RNA molecule that will be synthesized. So this is a process of splicing. And there are other complications are also involved in this process. For example, alternative splicing. So splicing is simply the removal of introns. But when we say alternative splicing, it means that alternative exons are joined together. So if you see a gene can, if you have heard that a gene can synthesize many proteins. So earlier it was a hypothesis by Biddle and Totem in which they performed experiment on Neurospora crassa, in which they said that one gene will code for one enzyme or one polypeptide. But later on, when it was observed that one gene is not only coding for one polypeptide, but one gene can code for many proteins. So how is it possible? So one of the process that is involved is alternative splicing. So what happens in alternative splicing? For example, there are three exons present in this precursor RNA. So they are joined alternatively. For example, if exon 2 is skipped and exon 1 and exon 3 are joined together. So in that way, there can be different isoforms of a particular gene could be formed. or we can say different transcript products will be synthesized. So that is alternative splicing, and we'll not go into the details.
Ma'am,
Yeah,
ma'am, what you just said,
isoforms, isoforms.
iso,
So the term isoform, why I didn't use it again, because the isoform is in the term of proteins. So when we talk about the protein products which are originating from a single gene, then we refer to them as isoforms, means the protein products originating from single gene. And when we call transcripts, which means the RNAs that are synthesizing from the same gene. So that is transcripts. That is at the level of RNAs, and in at the level of protein, we refer to them as isoforms. So yeah, different isoforms can originate from this alternative splicing process. So that's why if you talk about eukaryotes, then there is a huge diversity of proteins which are being synthesized. So that is due to this alternative splicing process as compared to the prokaryotes because they do not have introns in them. So, yeah, so now let's see this question. So, I hope this is clear to you about the splicing. Is there any doubt, or should I proceed to the question? Are you guys there?
Yes, ma'am, proceed.
Okay, okay, fine. So, now we'll be looking at the question. So, the splice site is found in dash. So, as I've discussed, that in introns the splice site is present. So, there are different options. So, what do you think is the right option? First is 3' end of the exon. Second option is 5' end of the intron. Third option is within the exon. And fourth option is within the intron. So, what do you think? Where is the splice site found in the RNA or the precursor RNA? We have already discussed the entire thing about the splicing, so I think it's quite clear what would be its right answer.
A,
Can
A,
anyone give
A,
A option?
A.
Okay, you
May
think
option B.
okay, B. Anyone else wants to answer? Okay. So so let's first talk about option A. So in option A, they are saying 3' end of the exon. So if we say splice site, so yeah, it is present after the 3' end of the exon at the beginning of the intron, at the border. So we cannot say that it will be 3' end of exon because it's not the 3' end, but after 3' end it's present. So we can say it is present at the 5' end of the exon. And it is not within the exon, it is not within the intron, but it is lying at the border and means it is not conserved in that exonic part. It is after that 3' end of the exon. So hence it is contained in the 5' end of the intron. Hence option B will be correct and not option A. So I hope it is clear. So now let's move to another question, that is the unessential components are excised off from the pre-mRNA strand with the help of splicing factors to generate the mature mRNA. Identify the component which is not part of the mature mRNA strand that has to be translated. So this also I've already discussed that what are the unessential components that are getting spliced out. So these
5'
are the
UTR.
okay. So you're saying option C?
Yes, ma'am.
ma'am,
A,
A.
A, A.
Okay, okay. Yeah. So it is, I mean, yeah, it is confusing that some people might think that option C would be correct because they are saying 5' UTR. So now let's see which option is correct. So it's option A, introns, because introns are getting spliced out during the splicing process. So intron is the correct. If we talk about exons, then exon are the most important components, so they cannot be excised out. So now if we see at the 5' UTRs, so UTR stands for untranslated regions. That means these will not be forming the protein, but these are not getting excised out because these are present in the RNA only. So I think you have the idea about the start codon and the stop codons, which governs that from where the protein synthesis will begin in the RNA. So 5' UTRs, they are the region because not means because in all the RNAs, AUG is the start codon. So it's not always the case that RNA will always begin with the AUG. Okay, so in certain cases, RNA can begin with some other sequence, and AUG could be present at some other site. So uh, that entire sequence till AUG will act as a 5' UTR, which will not get translated, but still it will be present in the RNA. So option C would not be correct, and option A would be correct. That is introns, which are getting spliced out. So now let's move to the main or the third part in which the whole expression is going to happen, that is the proteins. So how these proteins are synthesized? So first of all, the gene in A, you can say it's a genic segment which transcribes to RNA, and there is further RNA processing, and after that you will see a RNA molecule with the capping as well as tailing. So tailing in the form of polyadenyl tailing, poly A tail, and in the capping, the methyl group be attached to the guanine component at the 5' end of the RNA. Then it will translate to synthesize a polypeptide, or you can say the protein. So protein translation will always start at the AUG region. So in this B, if you see, there is a white region present towards the left. So after the cap, that particular region is the 5' UTR that is not getting translated. From AUG, the translation will begin, that is the protein synthesis. AUG codes for the methionine, that is the start codon, which codes for methionine. And then there are three different stop codons. So, is anyone having the idea about the stop codons? What are the stop codons? Have you read about it?
Yes, ma'am.
UAG.
Yeah,
UAA, UAG, or UGA.
yeah,
Those three
very
are stop code.
Yeah, right. So those are the three stop codons. So whenever any one of them comes, then the process of translation will stop there. So in this, you can see the C represents the polypeptide or the protein segment that is translated from this RNA. And then further, this protein also undergoes different post-translation modifications, such as in certain cases, methionine is removed. And then there are different things that can happen. Different functional groups can attach, methylation can occur, polyacetylation, different things can occur because the cellular machinery is very complex. So we'll not go into the details. So till here. So, one more thing that you must know is that the codons, these are triplet in nature. So, means three nucleotides constitutes a codon. When three nucleotides are present, then they will synthesize a single amino acid. So, there is a table which is being designed from where we can know that what kind of amino acid will be synthesized from these triplet codons. So, this is the table. I hope this has been discussed in the lecture. So, whenever this particular combination of the trinucleotides or you can say codons are present, then these particular amino acids will be synthesized. So this is the table, and you can see here there are different features of this genetic code. So out of which one feature is that a single codon can only code for a single amino acid, but single amino acid can be coded by more than one codons. So you can see clearly, for example, if you focus on this leucine, then it is coded by four different codons in this second columns, I mean second row, first column. So this leucine is being coded by four different codons. So in this way, this is a table, that is genetic code table, from where we can see that if particular RNA segment is given, then you can know that what would be its uh, protein or amino acid sequence. So now here is another question which says an mRNA strand consists of 120 nucleotides. Upon translation, this mRNA strand would form a polypeptide chain that is. So in this, they are saying that this mRNA strand constitutes of 120 nucleotides. So as it is related to that triplet codon concept, so what do you think would be the length of the amino acid chain? So how long amino acids will be
Option D.
synthesized? Option D,
Option
40.
D.
Okay, okay. Yeah, so option D will be correct because you know that three nucleotides altogether synthesize a single amino acid. So if there are 120 nucleotides, then there will be 40 amino acids. If we consider that the last nucleotides are not the stop codons and they are also coding for amino acid. As there are no other options was present, so 40 will be the right answer.
Madam, must be
Yeah.
word maximum in the question. Upon translation, this mRNA strand could form a polypeptide chain which is maximum 40 amino acids. It can be less than 40 also. No?
Yeah, right. Yeah, so there is one word missing, that is at max. What could be the maximum possible amino acid length? Yeah, so that must be present in this question. Yeah, but it's right. Okay, it can be shorter also. So now let's move to the another concept, that is mutations. So now we have discussed everything about the central dogma. I hope I have covered major topics and major questions. If you still have some doubt related to that, then you can tell me, and then we'll proceed to the mutations part. So is everything clear about the central dogma part?
Yes, madam. Let's meet.
Okay. So now let's move to the mutation part. So now what are mutations? So mutations, they can be of different types. One are point mutations and one are frame shift mutations. So mutations, you can say they are the changes or the variations that are occurring in the sequence. And this sequence variation is occurring at the level of DNA or RNA. And now, whether they'll be observed at the level of protein or not, it is a major question, because the certain mutations, there are different types of mutations. There are certain mutations which occur in the sequence, but they are not observed at the level of proteins. And why it is so? Because as you have seen the genetic code table, in which you have seen that more than one codon can code for single amino acid. And there is a hypothesis named as Vowel hypothesis. So even if a single nucleotide varies, but still it can code for a same amino acid. So hence, if we talk about point mutations, then as the name suggests, point, that means there will be a mutation at a single nucleotide region. So in this, even if a single nucleotide changes, it can lead to no effect at the level of proteins. So that type of mutation is known as silent mutations, which has no effect on the protein sequence. So in this, if you see this T that is getting mutated, so that is the uh, sixth nucleotide. So sixth nucleotide is getting changed from A to T. That is a mutation, point mutation, which is occurring, but still it is coding for the similar amino acid stretch, that is serine, valine, proline, tyrosine. So the amino acid stretch is conserved. So this is silent mutation, means mutation is occurring at the genome level, but you are not observing it at the level of protein, and it is silent, but still it is present. Then comes missense. As the name suggests, missense. So missense means there is some change in the sense or change in the sequence of amino acids. So in this, if you observe the seventh position, seventh nucleotide, so both the ends in one, C is getting changed to A, that is cytosine is getting converted to adenine. And what is the change at the level of proteins? So in this, amino acid proline is now getting converted to threonine, that is THR. I hope you are aware about these three-letter codes of these amino acids. So this THR means threonine. So in this missense mutation, there is a change in this amino acid sequence. So that is known as missense. Then there is third type of point mutation, that is nonsense. Non means there is no sense. That means due to change in a sequence, there will be the premature termination, or there will be the stop codon. So the sequence is changed in such a way that it is going to code for a stop codon. These sequences are written at the level of DNA. So don't get confused. If it is written TAG, that means UAG in terms of RNA. So UAG is a stop codon. Earlier there was TAC, which was coding for tyrosine, but due to the point mutation at the terminal region, means from C to G, now it is coding for UAG, and UAG is a stop codon. So hence this there will be premature termination of this protein whenever there will be the nonsense point mutation will occur. So now there are another mutations which are known as frame shift mutations. So in point mutation, the limit is that single nucleotide is getting mutated or changed. But in frame shift mutation, more than one nucleotide can change. There could be insertions or deletions. So in this, if you observe that the fifth and the sixth nucleotide are getting deleted, and there will be the change in the sequence of amino acids. So earlier it was coding for serine, valine, proline, tyrosine, and now due to the deletion of these two amino acids, now it is coding for serine, valine, leucine, and leucine. So frame is getting changed. So frame means there is the when we talk about frame, we means the sequence. in which order the sequence are lying. So if there is change in the order of sequence, so that is known as frame shift, in which the amino acid sequence will change. So this is the frame shift mutations. And now the question arises that how these mutations are occurring. So there are certain chemical agents, and there are also some of the replication machinery problems, because in replication also there can be, in certain cases there can be errors. Although the proofreading mechanism of our cell is very, means it's very good, it, and there is repairing process which is going on inside cell, but still there are certain cases where there is a leakage in the process of replication or transcription, in which um, different nucleotides get mutated and they get retained. They are not repaired, they are not proofread. So that could be the one reason that how mutations are coming and getting expressed. And another reason is the mutagenic agents. Due to certain chemicals, due to radiations, these mutations are getting observed. So, for example, in this case, if you see the deamination of the uracil or the deaminated cytosine can form the uracil, due to which in one strand it will form, the G will form bond with the C, but in another strand, as C is deaminated, so U will bond to A. So in this way, the mutation is getting observed in the top one DNA duplex, if you see. So that is due to the deamination of cytosine. So whenever deamination of cytosine will occur, then it will form uracil, because these nitrogenous bases are almost similar to one another except for the functional groups that are present on them. So whenever deamination of cytosine occurs, it converts to uracil, and uracil will bind to adenine. So in both the DNA duplexes which are newly synthesized, they will have a change in the sequence. And further on, this mutation will be carry forward, because as the mutated DNA will get replicated, so this mutation will get established in the further generations. Similarly, the depurinated adenine, that means adenine is removed. So if adenine is skipped, then this thymine in one strand, it will be converted, it will be bounding with the adenine, but in the another strand, it will not be able to bound, and then C, then this particular A is skipped. And so in this way, in the mutated, you can see that there is missing of this adenine and adenine thymine bond. So in this way, mutations occurs inside the DNA. So this is the entire process of mutations. So is it clear? Do you want me to repeat something, or do you have any doubts about this? So is there any doubt?
No, ma'am.
Okay, so if there is no doubt, then let me proceed to the question. So which one of the following mutation types is not likely to generate a truncated protein? So this thing I have discussed during explaining you the process of mutation. So first one is missense mutation. Second option is frameshift mutation. Third option is nonsense mutation. And fourth option is point mutation. So truncated protein means that if particular mutation will occur, then the protein which is earlier coding for this stretch of amino acids, now it will not code for the same stretch of amino acid. It will truncate or stop in the in between only, where that mutation is occurring. So what do you think? What would be the right answer? Which kind of mutation is not likely to generate a truncated protein? So truncated protein means
Option C, nonsense
option
mutation.
nonsense mutation. Okay. So any other wants to guess?
Ma'am, missense.
Missense. Okay. Anyone else?
Ma'am, truncated means breakage in from middle.
From any part where the particular mutation is occurring. So truncation means that if particular amino acids are getting coded, so then the stop codon will be coded due to that mutation. The amino acid will, means the earlier amino acid will not get coded, and due to mutation, there will be the formation of
Nonsense.
stop codon.
Nonsense mutation. Nonsense mutation.
So the question is that which of the following mutation type is not likely to generate a truncated protein?
Okay. Not
Yeah,
not likely.
yeah, not likely. So what do you think? Which option? Somebody said option A, somebody said option C.
Mis-
Okay, so
Missense mutation. Missense mutation.
yeah, right. So the correct answer will be the missense mutation, because what happens in missense? due to the mutation, there is change in the amino acid sequence. So if, for example, if leucine is getting coded in the earlier from the earlier transcript, then now in place of leucine, isoleucine will be coded. So that means there is missense, change in the amino acid that was earlier getting coded. So when we talk about frame shift mutation, so in certain cases, due to frame shift, there could be the possibility that it could get truncated earlier. There could be the formation of stop codon. And if we look at the nonsense mutation, then it is the mutation in which the protein get truncated, because in this, due to this mutation, stop codon will be expressed. Similarly, in point mutation, if we talk about the nonsense category of point mutation, so it will also generate the truncated protein. So in all of these, the missense mutation will be the correct option, which is not likely to generate a truncated protein. Okay, so let's move. So the next concept is the chromosomal abnormalities. So now mutation part is covered. Now we will look at the chromosomal abnormalities. So do you have any doubt about mutations, or should we just proceed with these chromosomes?
Madam, last question.
Yeah, okay.
Um, Truncated means half protein, no?
Truncated means it doesn't mean
Not a
half.
complete protein.
Yeah,
Or
truncated
it is
means
not
not a, yeah,
complete protein.
yeah, not complete protein.
Point mutation will also produce complete protein. Frame shift will also produce complete protein. Even missense will produce also complete protein. Only there is stop codon in nonsense mutation.
So the question is that which one is not likely to generate. So nonsense will always generate a truncated protein, right?
Protein.
So in,
Other
yeah,
three will generate normal protein. So missense mutation will also generate complete protein. Though it is not correct protein, but it will generate complete protein.
yeah. So the question is that which of them will never generate a truncated protein? So missense,
Point mutation. A,
so
silent point mutation also will never generate truncated protein.
but the option here is that out of them, which could never. So because in point mutation there are different categories as well. So in point mutation
Uh, Point
means,
mutation, there are three categories in point
yeah.
mutation.
So in missense mutation is also one among the point mutation only, right? So in this they are talking that 100% you can say. So in missense mutation we are 100% sure that it
Missense is
will, what missense is,
missense can be conservative or non-conservative.
yeah, it
It can produce
actually,
or it cannot produce.
but missense will never truncate a protein. The question here is that which one will generate a truncated protein. So the name
Not
missense,
not
we are not,
even frame shift mutation can generate normal protein. It will also not generate. Even point mutation cannot generate truncated protein.
actually
But at
the,
point mutation, silent point mutation won't generate a truncated protein. Correct?
yeah, but in this other point mutations,
Not
so
only that, it will generate a near normal protein.
so when they have given a broader term that is point mutation, so we have to keep all other types in mind. So means we are not sure which kind of point mutation they are talking. So in this we have to choose only that option where we are 100% sure. You are right. Then in point mutation there are certain other mutations, type of mutations which are also not likely to generate a truncated protein, such as silent. And missense is also one among the point mutation only. But still, if we are given point mutation, we are not clear which kind of point mutation they are talking. So out of
Madam,
these, yeah,
sorry to interfere, but what I understand till now is कि
yeah,
whenever there is stop codon, then only the protein production will stop.
right,
And stop codon will be produced in the nonsense mutation only.
yeah, only in the non,
Only C will generate a truncated protein. So all three other options can generate a protein that is not truncated.
yeah. But in frame shift mutation, there could be a possibility because frame shift means insertions or deletions. For example, if a
Pos-
particular insertion
yes,
comes,
madam. There is still possibility it will
yeah,
produce a stop codon and there will be truncated protein.
yeah,
Okay.
yeah,
My confusion is with point mutation.
yeah, your confusion is with point mutation. So I'm saying that in this they have mentioned point mutation only. So if they have mentioned a particular type, then we could say that point mutation is also not likely to generate a truncated protein. But if it's a broader term given, so we cannot select point mutation because the category of point mutation, which is nonsense mutation, it can lead to generation of a truncated protein. So we cannot go with the point mutation, right? Because it has three different types.
Maybe my knowledge is
No, your knowledge is
very less, very less. But I will try to
it's
read
good.
and
No, no, no. You are
understand.
right. You are right about point mutations. There are three categories of point mutations. But why we are not selecting point mutation? Because in point mutation, nonsense mutation is also one category in which it could generate a truncated protein. Because in point,
Missense protein will be also generate truncated protein.
why we call it as missense mutation? A point mutation will always be called as missense mutation when it is going to change the amino acid sequence. If it is going to stop it, then it will be named as the nonsense mutation. So point mutation is a broad term. And then we
Suppose
name it on the basis of the outcome, that what would be the name of that particular point mutation. Missense mutation is nothing, it is a point mutation only. But why we are
okay,
regarding it as a missense? Because it is going to change the amino acid sequence. So that's why it
no,
is,
but
yeah.
it will generate complete protein. No?
Yeah, it will generate a complete protein and it will not
Example is sickle cell disease. No?
the sickle cell anemia. Yeah, that is a,
Instead of
yeah,
glutamic acid, there is a valine, but still that protein is complete. Hemoglobin protein is complete.
yeah.
Amino acid chain is 141 amino acid long. It
Yeah,
is not truncated.
yeah, it
That's
is
what
not
I am
trunk.
saying.
Yeah, so the question is also the same, which is not likely to generate a truncated protein. And we are saying that missense mutation is not
Missense,
going to generate.
but sickle cell anemia is
Ma'am,
also a
I
point
think Sal
mutation.
is confused between truncated and distorted protein.
Okay,
Truncated
but
means, according to Google,
yeah,
truncated mean to make something shorter, especially by cutting off from the top or end.
okay. So,
So only missense mutation will be the answer
Can be
due to
distorted
the
protein.
shorter length. Due
Suppose
to
D
the
so
shorter
option
length.
would have been silent point mutation. Then which will we choose?
yes, then if it would have been mentioned silent point mutation, then it would be confusing. Then they might have given A and B both. So in this, they have mentioned the point mutation. So we are not sure that what kind of point mutation because they haven't named it. So there is still possibility that if nonsense point mutation will be there, then it will not. Means then there will be a truncated protein.
Nonsense is surely not an option, but other things, same shift also we can get a complete protein. Okay, madam.
I think you
Whatever.
are getting confused with the question. I think because the
Sorry.
question is not likely. Yeah, it's fine. I hope you got the clarification. If not, then we can further discuss it somewhere else because we need to proceed further without wasting any time. So I hope it is clear about the mutation. Now we'll be focusing on the chromosomal abnormalities. So when we talk about the chromosome, then there are different abnormalities which are involved with this, due to which different disorders we observe at the level of phenotypes. So there are two kinds of abnormalities. One is at the level of numbers, another one is at the level of structures. So numerical level means if there is so when we talk about chromosomes, then there are two kind of chromosomes. One are autosomes, another one are the sex chromosomes. So when we talk about the sex chromosomes, and they are X and Y, and all other 22 autosomes are referred to as somatic chromosomes. So in on the basis of numbers, what kind of abnormalities can occur? That is trisomy. That if particular chromosome is present as a triplet, then it is trisomy, or it is monosomy because we know that chromosomes are present in the form of pairs. So if that pair is disturbed due to non-disjunction or any other process, so there will be trisomy. or monosomy, that instead of a pair, one only single chromosome is present and another one is missing. So that is monosomy, or triploidy. So when we talk about ploidy, that means sets. So three sets are present. So this is the at the level of numbers, and then at the level of structures, that means there are deletions in a chromosome or duplication in a chromosome. Particular segment inside that chromosome can duplicate. There could be translocations, that one segment of the chromosome could translocate to some other chromosome. There could be inversions, that a particular due to double strand breaks, a particular segment gets separated from the chromosome, it get inverted, and then it get again inserted. So that is inversions. Or the isochromosomes, which are almost similar, then dicentric chromosomes, where there are two centromeres, and then the ring chromosomes, in which there are double strand breaks, and then the remaining part of the chromosome, it forms a ring, it joins back. So these are different abnormalities which occurs at the level of chromosomes. So, a few examples we can discuss, because these are most important abnormalities which are majorly discussed whenever the questions are asked. So, first one is Down syndrome, which you have heard a lot of times. So, it occurs due to the trisomy of 21 chromosome. Whenever an extra copy of chromosome 21 is present, then this is at the level of numerical chromosomal aberrations, because a single copy is present as an extra. So, there will be 47 chromosomes. Otherwise, there are 22 autosomes along with the 6 chromosomes, and then they are existing in the pairs. So there will be 46. But in this, as there is trisomy of chromosome 21, so there will be 47 chromosomes will be present in the total. And it can also occur due to the translocations. So in certain cases, the chromosome 14 and 21 can show the translocations of the components, due to which the Down syndromes can be observed. And then there is a certain term which is a mosaic type. So mosaic type means in two different cells, there are two different types of chromosomal, or the karyotype is observed. So in one cell, you observe the 46XX, and in another, you observe 47 with XX along with the 21 extra copy is present. So this is the Down syndrome. But mostly whenever we say about Down syndrome, then we always talk about the trisomy of chromosome 21. Then comes the Turner syndrome, in which you observe the monosomy of X chromosomes. So it is generally observed in the males where they lack the Y chromosome. So in them, the features will vary. So that is the Turner syndrome. Then comes the Linfelter syndrome, which we will heard again and again. So in this, there will be an extra copy of X chromosome that would be present in a male, due to which that male will show certain features of the females. So that is due to the extra copy of X chromosome. Then there are certain other kind of syndromes which you will observe due to the change in the number of chromosomes, or due to the structural variations of these chromosomes. So these are further mentioned in the table. You can see them whenever I'll give you this presentation. This is just for understanding. And then in here also, in the translocations also, you can see the chronic myeloid leukemia, in which there is translocation of the chromosome 9 and 22. This why I'm discussing this, because in the questions you'll observe that this is a major point of discuss, because in the chronic myeloid leukemia there is a term associated with this. This translocation is very important, so that we will discuss later. And in deletions also, you can see that there are certain fragments which are getting deleted and leading to certain cancers or anemia or meningioma. So there are different abnormalities which are occurring due to the changes at the chromosomes, both at numerical level and structural level. So now let's observe this question, which is
Excuse
given.
me.
Yeah, yeah, right. Say.
Uh, I am not understanding from the uploaded videos for imprinting disorder.
Yeah, so we will discuss them after this. After chromosomal abnormalities, we will discuss that also. Imprinting disorders, what is chromosomal imprinting or genomic imprinting? So we will discuss that. I hope
Okay.
we have time. So yeah, I will discuss that. Don't worry.
Okay.
Yeah. So this is the question which says that given below is the karyotype of a patient suffering from a congenital hernia. Examine and identify the option that lists the correct karyotype and cause for the defect. So I hope you know what is a karyotype, right? Karyotype means for an individual, the entire chromosomal set which is present, we capture that what are the chromosomes present. So in this question, they are saying this is a karyotype of a person who is suffering from congenital hernia. So now observe this karyotype and say that what do you observe? So where is the defect which is causing this hernia? So the options are there are 46 chromosomes, X and Y, but there is some deletion in some chromosome. Then the option B says that there are 46 chromosomes and XX is present, and then there is some deletion. Similarly, option C is saying 46 chromosomes are present, there are XY chromosomes, and translocation is there. And then option D says the 46 chromosomes are present, then XY chromosomes are present, and there is some inversion. So observe this karyotype. Can you answer what kind of defect is there?
Name A.
Option A.
46XY deletion.
Okay. Yeah, so now let's see what kind of defect is there. So yeah, it is 46XY deletion because you can see all the chromosomes are present. But if you see this 15th chromosome, so it's one pair, it is showing the ring chromosome formation. So how the ring chromosome is formed, as I have discussed earlier also, when there are two breaks, two strand breaks are there. So from both the ends of that particular chromosome, there are breaks, and then the remaining fragment, it folds and it fuses and it forms a ring. So which you are observing at the chromosome 15th. So yeah, it's right. There is a deletion at the chromosome 15. So now I just want to give you a brief information about this hernia. So what is congenital hernia? So hernia is a disease which occurs when an internal part of the body it pushes through the weaker muscles and its surrounding walls. So due to which it forms a lump. You can observe a lump due to the weakness in the muscles. The particular body part it get folded to that part and means there is some you can say a projection from the body. It can occur at different parts of the body, and on the basis of that, different terms are given to hernia. Different means sometimes it's called as diaphragmatic hernia. So in the right side, I'm showing a case where there is diaphragmatic hernia. So in which what happens that this small intestine is getting pushed. It's compressing the left lung and it is getting pushed towards the left lung side. So this is called hernia. Means whenever there is weakness in the muscles, then certain parts protrudes to some other region only. It's pushed towards that site, and they are causing some kind of a lump, or you can say projection. So that is hernia. And congenital means
Madam,
yeah,
hernia is defined as abnormal things they are passing through a normal opening.
yeah, right.
Abnormal content that is passing. The diaphragm is not made for intestine to go up, but intestine go up. It is for coming off esophagus. Same way in the spermatic cord is for that hernial site at a thigh. But suppose abnormal
Yeah,
thing or intestine go there, that becomes so abnormal content in a normal opening. It means hernia.
yeah, right. Yeah, right.
Simple.
Yeah,
Madam, one
simple.
more thing.
Hmm.
In the question, another word I came across is karyogram.
Okay,
What is
so
different? What is the
karyogram,
difference between?
yeah,
I think doing is karyotype and what we get is karyogram.
right, you're right. So means when you observe
So,
that film, so that is the graph which you see is the karyogram, and karyotype is what is the
so again
nature.
there
Yeah.
must be again question correction in the question. Given below is the karyogram.
So and they are saying that given below is the karyotype of a patient. So in this, they're not saying about the karyogram. Karyogram is the thing which we are seeing. But if we talk about the patient, then the property of a patient is the karyotype. So means
But,
yeah. So
but
in this,
in the answer they had given the karyogram. There is written karyogram. The
yeah.
karyogram descriptor deletion. Whatever we are seeing, it is a karyogram or karyotype. That's
So what you see is a karyogram. Okay. But if we say
and what we
patient for patient, so it is a karyotype of a patient. So are you getting it? It means that karyotype is a property of a patient. That karyotype means what are the chromosomes present. But where you are seeing that karyotype, that is in the form of karyogram. So when in the question it's mentioned karyotype of a patient. So it means that patient's karyotype, what are the chromosomes are present. But in the answer they are mentioning about the karyogram. So means
what my knowledge goes is arranging the chromosome in order from according to their length is karyotyping. and what we get is karyogram. Means this
Yeah,
is
you
new
are
word
right.
for me.
You're right. So, for example, if you say about the electrocardiogram which you get for a patient, so
ECG.
ECG
The
of a
graphy
patient.
is doing it, and what we get is a electrocardiogram.
Yeah, right. So
So
means
it is
in this,
I think it is karyogram. I think whatever.
yeah, whatever means you can write karyogram also, but why they've mentioned karyotype? Because they are talking about the patient, that karyotype of a patient. But yeah, they can write karyogram also because the thing which we obtained is a karyogram, means in which we obtain the karyotype is a karyogram. So, okay. So now let's move to another question. So the question is, which one of the following options correctly matches the syndrome to their respective karyotype? So earlier we have discussed few of the syndromes and their karyotype. So first option is Down syndrome with 47 chromosomes and XXY. Option B is Turner syndrome with 47 chromosomes XY plus 21. That means additional copy of chromosome 21. Then the option C is Linfelter syndrome with 47 chromosomes and XXY. Then option D is Linfelter syndrome with 45 chromosomes with X and missing Y chromosome, that is XO. So what do you think is the
Ma'am,
correctly matched? Okay, option C.
Yes, ma'am. Option C.
Okay, right. So in Linfelter syndrome, as I've discussed earlier, there is one extra copy of X chromosome which is present, and rest others and that are incorrect, because in Turner syndrome they say 21. That is, 21 trisomy is observed in Down syndrome, not in Turner syndrome. So it's incorrect. So in Linfelter syndrome, if you observe the karyotype, then you'll see there are two X chromosomes present, and these are few features or symptoms you can see. Means whenever you see a patient with the Linfelter syndrome, so these are the things you will notice. So they have certain features which are lesser like males and more like females. So that's why it is Linfelter syndrome. So now there is another question. So
Madam, again,
yeah,
sorry.
yeah, it's okay.
Can we get XYY also?
XYY means till now I haven't seen that case because
Is there possibility?
I don't think there is a possibility, because in female there are two XX chromosomes and male there are one X and one Y. So
Madam,
means,
we get four gametes now, two X and two Y. If there is a disjunction, we can get XYY.
but as per my understanding, I don't think you will get two Y because male and
Okay, madam.
female.
Please proceed. Just a
Yeah,
confusion, that's why.
yeah. But means I haven't means come across any case with two Y chromosomes. So now next question is, identify the options that correctly fill in the phenomena described in the statement given below. The Philadelphia chromosome that causes chronic myeloid leukemia is an abnormally short chromosome 22 and is a product of dash event. So,
translocation.
Translocation.
okay, very nice. You guys have idea. So yeah, so earlier I was discussing about that chronic myeloid leukemia because there is a this term associated, that is Philadelphia chromosome. So yeah, you are right. There is a translocation in which you see that there are two chromosomes. The translocation occurs between a normal chromosome 9 and chromosome 22. So normally the chromosome 22 is very small as compared to chromosome 9. So what happens? The segment of the chromosome 9, it breaks and it gets translocated to the chromosome 22. Whereas the chromosome 22's the longer arm break take place and that get translocated to chromosome number 9. So in this, the chromosome 22, which get changed, is referred to as Philadelphia chromosome, which has now two genes, that is BCR as well as ABL. With this translocation, and this chronic myeloid leukemia, it is a kind of a cancer in which it occurs at the white blood cells. So what happens is that the BCR and ABL genes, when they join together, then they lead to the formation of a mutated protein, that is tyrosine kinase, which help this gene to divide indefinitely, because in cancers there is a continuous cell division and uncontrolled cell division. So this translocation is helping it for the uncontrolled cell division, due to which it is causing the cancer, or you can say leukemia. So this is one thing you must know about this translocation, that what is the mechanism for this leukemia. So if everything is clear, then I'll proceed to the another thing, that is the genomic imprinting, as somebody was asking about that. So should we proceed to the imprinting part if everything is clear about these aberrations, chromosomal abnormalities? Okay. If there are no doubts, then let me first check that are you guys there? Yeah, so should I proceed?
They will block
Ma'am,
you,
I have a
ma'am.
question.
You will block me.
No, no, no. You can ask as many doubts because this is the doubt session, so nobody will stop you from asking questions. And in this way,
Because
you
in
will,
chronic
yeah.
myeloid leukemia, they especially ask karyotype person to do that Philadelphia chromosome. So
Yeah,
I had heard about the spectral karyotyping. So
right.
22 and 9, they are in the nucleus when they are compact, they are nearby chromosomes. Why the from 9 to 22 means for arrangement it is long, means there is a gap of 11 chromosomes or 12 chromosomes is there. But are they are residing side by side, because to translocate from one chromosome to another, they must have been
Yeah, you are,
nearby.
yeah, you are right. So in cell, they means na cell is not that big, right? It's very means in that chromosome are very compactly packed inside the nucleus. So for this translocation, they're not that far away from one another. So means I haven't observed means under the microscope or anywhere where I could see the positions of them, but yeah, they're not that far. Means
In one lecture, I had seen that. I think that is near. My means I had
yeah, they
not
are
seen,
nearby. Yeah,
but I
because
think it is near to the 22. That's why the
yeah,
translocation
yeah,
occurs
exactly.
from 9
Yeah,
to
so they
22.
are
Because
nearby to one another.
or other translocation, they are not fit for life. That's why they
Hmm.
are not there.
Yeah,
That should be there.
yeah, that should be the answer.
Thank you,
Yeah,
madam.
they are residing. Yeah.
Thank you,
Yeah,
madam, for
and
your.
one thing I want to mention because I've seen your assignment. So in that there are two terms. One is spectral karyotyping and one is standard karyotyping. So what spectral karyotyping means, as the name suggests, spectral. That means spectra. That means colors. So whenever the karyotyping is done in the form of colors, with the colors you are going to distinguish between different karyotypes. So that is known as spectral karyotyping. Means with the extent of color, you are going to observe the karyotypes. So that is spectral karyotyping. So now let's discuss about the genomic imprinting. So genomic imprinting. So printing means you're going to print something. So imprinting means if there is not correct printing. Printing means genes are forming proteins. That is printing from gene to protein. So imprinting means if gene is not forming protein. So imprinting means that the gene is silenced or gene from the other parent is expressed. So in what happens in genomic imprinting, that only the gene from one of the parent is getting expressed and another one is getting repressed or not expressed. And now also its mechanism is not completely understood that how it happens, but there are certain studies which says that there are some epigenetic modifications that are occurring at the gene level which are causing the uh, certain genes to not express. Yeah, genes coming from one parent is not getting expressed. So this process of silencing the genes through the process of DNA methylation is termed as genomic imprinting. So in this, the repressed allele is methylated while the activated allele is unmethylated. So whenever anything gets methylated, so its expression gets repressed. So you'll observe, for example, you also know about the X chromosome inactivation. So in that also, this methylation happens. And I think you know about the heterochromatin and euchromatin also, because when we talk about chromosomes, then there are two types. One is euchromatin form, one is heterochromatin form. So euchromatin form is the form that is coding for the protein, whereas heterochromatin form is the one in which the chromosome is compactly arranged, in which it is not able to code for the protein. So that is heterochromatin form, and to support that heterochromatin form, the methylations is occurring. So methylation leads to DNA inactivation, or you can say gene inactivation, in which it is not allowed to form the protein. Similarly, in imprinting also, whenever one copy of the gene gets methylated, so it is not going to express. And most of the cases in imprinting, only the one parent is going to express the protein, and from another parent, there would be the genomic imprinting, and hence you will not be able to observe the protein. And there are certain examples of this genomic imprinting as well. One is Prader-Willi syndrome, and another one is Angelman syndrome. So in both of them, there is the imprinting or the other errors which are occurring at the long arm of the chromosome 15. So in the right side, you can see the first top one shows the non-imprinted genes, in which from both male and female, the genes are coming and they are getting expressed, they are getting transcribed. But if there is imprinting at the level of, so if there is maternal imprinting, that in mother gene get imprinted, there is some kind of methylation, so it is not going to express, and only the male is going to express that gene. So that is known as genomic imprinting, in which only the sex specific gene is getting expressed and another gene is getting repressed or unexpressed. So now let's see a question which will make
Ma'am,
you, yeah.
ma'am, is this imprinting disorder caused due to loss of function mutation? And what is actually meant by loss of function mutation?
So loss of, when we talk about mutations, then in mutations we are mostly talking about the nucleotide sequence, that the changes are occurring at the level of nucleotides. So then we call it as mutations. And due to mutations, if there is, means earlier it is going to code for a protein which has some function, then now due to mutation, the sequence has changed and now different amino acid has been synthesized. So now they're not going to perform the same function, or they have lost their function which they are performing earlier due to the change in the nucleotide in the sequence. But when we talk about imprinting, then it means the nucleotides are exactly the same, but there are certain other agents. They are epigenetic agents. So epigenetic means other than the normal agents, the nuclear agents, the external agents when they are acting, they are known as epigenetic agents, means which are lying in the environment of that gene. So that is not a kind of a, means we can't say it as a mutation exactly. So in this, what happens is the another proteins are coming and they are methylating a particular nucleotide, and in that way they are leading to the suppression in the expression, but the nucleotides are same. So uh, we cannot say it as a loss of function mutation exactly. Means either, means yeah, there is the loss of function because now it is not going to code, but I'm not sure that whether we'll be regarding it as a mutation or not, because in this there is, means not, nothing is getting changed at the level of nucleotide sequence. There is a modification of the nucleotides. So I need to see this. Then I can say that either it will be regarded as a loss of function mutation or not.
Okay, ma'am.
Yeah, but means you can see that there are some epigenetic factors that are involved which are causing this imprinting, and this is also sex specific. And yeah, there is loss of function, but for the mutation, I'm a bit confused that either you'll call it as a mutation or not. But for now, I think, means as per assignment, I guess you can say it as, means I'll confirm it, but maybe it could be a loss of function mutation, but I'm not sure. So I'll tell you in the next session that what it is. Yeah, so this is a question in which you'll be get a better understanding of this genomic imprinting. So there is a disease which I told, that Prader-Willi syndrome, which results from the different molecular mechanisms. So in this, they have given this, after DNA fingerprinting, you observe the polymorphic, means by using different restriction fragments, you observe this graph which you get after the digestion. So they have said that you have a patient that you suspect has this disorder, that is Prader-Willi syndrome, and you send the blood samples of the patient and of both the parents of that patient to the lab for genotyping using a DNA polymorphic marker located close to the centromeric region on the chromosome 15. Because we have mentioned earlier that Prader-Willi syndrome, it is going to occur at the chromosome 15. So the following genotypes are found. What is the most likely interpretation of this disease? So in this, the F marks the father, P marks the patient, and M marks the mother. And these are the bands which are observed after this polymorphic marker and the digestions which have been performed. So These are the options. Option A is a new mutation in the gametes contributed by the father. Option B, paternal deletion. Option C, laboratory error. Option D, non-paternity. And option E, maternal uniparental disomy. So now observe this graph and tell that what do you think? What are your interpretations? That what has
Maternal
happened?
uniparental disomy.
Yeah, anyone else?
See,
Okay. So in this, the correct answer is the maternal uniparental disomy. And why? Because when we observe the patient and the mother, then we see that exactly same bands you observe for both the patient and mother. But whereas in father, means the 3 and 4, you do not observe in the patient, which means that this there is imprinting of this. So there is paternal imprinting. That means the paternal genes are not getting expressed, they are getting imprinted. So which means the maternal uniparental disomy. That means the mother is the only parent where you observe this disomy. So that's why its correct answer is maternal uniparental disomy, because the other parental genes are repressed for this chromosome 15. Yeah, it's right. Yeah, so this was all about the week one, and I hope I have covered the almost the most of the topics which you might have encountered in your assignment for the week one. So yeah, there is still some time, so we can also discuss some of the content of the week two. So do you have any doubts with this week one content and about everything that we have discussed till now?
Ma'am, I have a question, not doubt.
Yeah, okay, you can ask.
Can you please tell me something about non-allelic recombination?
Known allelic recombination. Is it right?
Yes, ma'am.
Okay, so when we talk about the recombination, so which means there are two means allelic means alternate form of a single gene is present. Okay, so when we say allelic recombination, so we mean that similar form of a gene is getting recombined. There is a recombination between the two homologous chromosomes which are carrying that allele. But when we are saying non-allelic, which means that one alternate form of a gene, and then in another chromosome there is some another form of gene. So which means these both are not alleles. And when there is some recombination is occurring, so that is known as non-allelic recombinations. Means the two chromosomes, or you can say two sister chromatids, are showing the recombination where both of the genes are not alleles but they are non-allelic. Means they are different gene different forms of the gene. They're different alleles, you
Okay,
can say.
ma'am. So, due to non-allelic recombination,
Hmm.
one chromosome will have both gene, both allele A or B.
Yeah.
The another one which involved in the recombination is got deleted.
Yeah, that could also be the case. Means for example, as you are saying that the chromosomes are not homologous chromosomes, they are different because there is non-allelic recombination. So if in case in one chromosome you have one gene, in another you have two genes, so after recombination, for example, that segment get transferred to the another chromosome, so that particular gene will be missing in that particular chromosome from which it was earlier present. So yeah, it could lead to the deletion, or you can say, I mean, deletion of that particular allele from that region.
Yes, ma'am. I
It could
am
get,
just asking
yeah,
that.
yeah. So that could also be the case in which instead of means one allele get missed from one chromosome and it could transfer to the another. Okay, so is everything clear? So should we discuss about the week two also? Little bit of week two content, if you want.
Hello.
Hello? Yeah?
Yes, ma'am. I have some doubt regarding the imprinting disorder. As you said, the imprinting disorder is like specific to the sex specific. Means what does it stands for? Does it means the
Yeah.
imprinted in case of imprinted male disorder? So is it going to be inherited to the male patients, or is it going to be inherited in both sexes of the offspring, in both the gender?
So when we say imprinting, we do not mention about the offspring. We talk about its parents. So because each offspring is getting the similar contents from both mother and father, so imprinting is occurring at the level of parental level. Means so if it's a father's or if it is the paternal imprinting, so that means the genes which are coming from father, they are not going to get expressed, whereas the genes coming from mother, they are getting expressed. So means we are not here referring to the gender or the sex of the offspring. That could be either male or either female. So imprinting could be observed in both the male and female. Because when we say imprinting, so we are not exactly saying that it is of the sex chromosomes, right? We are saying all the chromosomes. Imprinting can occur at different chromosomes. For example, in Prader-Willi, we mentioned that it is the chromosome 15, so which is present both in male and female. Yeah, so yeah, it is not based on the gender of the offspring.
So like
Yeah.
that you have said that the Prader-Willi syndrome
Hmm.
is located due to some problem in the chromosome 15. So
Hmm.
that's an autosome, right?
Yeah,
So
that is
so
autism.
in case of Angelman syndrome, so in that on that disease, so how it's going to be the inheritance of or the means how that imprinted genes are getting inherited to the next generation.
So, if we talk about the Prader-Willi and Angelman syndrome, then they both are occurring at the chromosome 15, but the difference which lies in both of them is that in Prader-Willi, it is the paternal chromosomal imprinting of the chromosome 15, whereas in Angelman, it is the maternal imprinting of the chromosome 15, in which the mother chromosome 15 is getting repressed. So, the imprinted copy is getting transferred to the offspring as it lies, it is a methylated form is getting transferred. Means you mean that gene is not getting expressed, but still it is present because transfer is still happening, right? Means
Yes.
yeah, so it will get inherited to the offspring, but yeah, it will not express because it is methylated, so it is in the imprinted form, right?
So, so in case of Prader-Willi syndrome, you said that uniparental disomy, maternal uniparental disomy. So, am I correct that if in case of Angelman syndrome, it would be imprinted, maternal imprinted, and uniparental parental, I think parental uniparental disomy.
Paternal uniparental
Yeah.
disomy. Yeah, right.
So, is it fine?
Yeah, it's
Uh,
fine.
yeah, yeah.
Yeah, okay. So, any other questions? So, what are your views regarding the week two content? So, should we discuss it or should we just
It would be extremely nice if you can discuss few of the problems, analytical problems regarding the second.
okay? So, means I have listed certain uh, problems which are related to the means which are just week one first content, which was about the Mendelian patterns. So, I can discuss that. Let me show you.
Only 6 minutes are left, madam. Only you can please tell
Yeah,
problem.
right, right. So let me share. So is my screen visible?
Yeah.
Yeah. Okay. So in week two, uh, till now, means as of now, I'm just going to discuss with you the Mendelian pedigree patterns. So I hope you have the idea about this pedigree symbols. If you are means you have the idea about this, then we can simply skip to the problems. We'll not just go in the detail of this. And you have also read about this five types of Mendelian pedigree patterns. That is autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and then Y-linked. So these are the pedigree which tells about the ancestry and the lineage of a person. So let's quickly look at the problems. But before that, you must have the idea about what is autosomal dominant, what is autosomal recessive, what is X-linked recessive, X-linked dominant, Y-linked. So let's see the problem. If you will not be having the idea about this, then we'll just go back and study them. So the question is that Sonali and her husband Ramesh are phenotypically normal, while Sonali's father was affected with an X-linked syndactyly condition called webbed toes. Sonali's first child, Rohan, is also affected by the X-linked phenotype. Sonali and Ramesh are expecting their second child. So what is the probability that this second child will have the syndactyly condition? So these are the options. So can you guess what kind of the pedigree pattern is occurring in this? In which they are saying that the Sonali and her husband they are normal, but Sonali's father was affected with some X-linked syndactyly condition. So here they have mentioned that what kind of pedigree pattern is there? That would be X-linked. And whenever you see X-linked father, and you see that the daughter is not showing the disease, that is phenotypically normal. So you should get this thing very clear that whenever a disease is X-linked dominant, so the fathers will always transfer it to a daughter because father is giving his X chromosome to the daughter. So if it would be X-linked dominant, then Sonali would have shown that particular syndactyly condition. So this is very clear that this particular pattern is X-linked recessive pattern, right? Is it clear that why it is X-linked recessive? Is it clear?
Ma'am, I will
Yeah.
Because why
answer.
would not be
Option
should
B will be the answer.
yes, ma'am, B.
Option B. Okay. So is it clear that why it is the X-linked recessive pattern of paternity pattern? Is it clear?
Yes, in mother
Yes,
it is
right?
not expressing
Yeah.
due to one heterozygosity.
Yeah, right. Heterozygosity and it is recessive. So
So this has always been the case in case of X-linked disorders. So it's hardly we can see some inheritance of like male offspring from the father. It is always the
hmm,
inheritance of the defective gene from father to daughters.
daughters
But in
because
case of
yeah.
the X-linked dominant,
Hmm.
so in case of X-linked dominant, it can only be inherited if mother is carrier and father is sorry, yeah, uh, no, that has been the case actually. It cannot be inherited by the sons from the father.
Yeah. Anything that is X-linked, so father will not transfer it to their male child because father is giving his Y chromosome to the male. So X-linked thing father cannot give to the male child. They will always give it to the female. And then it depends that either it is recessive or dominant. So if it is recessive, then father can transfer it to the daughter, and daughter will act as a carrier. If if that child's mother is normal, if she is also a carrier, then the daughter can have that disease. So, yeah, so now let's focus on this. So, in this, what is the probability that the second child will have the syndactyly condition? So, somebody mentioned the option B. So, its option will be that uh, 50% regardless of the sex means. So, what is the reason behind this is that this Sonali is the
There's a
okay,
thumb rule actually. It's already mentioned in thumb rule.
yeah, yeah. So means regardless of the sex, 50% will be having this syndactyly because Sonali is a carrier for this X-linked mutation because her father has given her that particular X-linked trait, and then since um, Sonali's father as well as her first child are affected by the disorder, so hence every child, regardless of the sex, will carry this 50% probability of receiving the mutant X chromosome from Sonali, and hence the probability of disease is 50%. Okay, so now let's move to the next question. Is it clear why its answer is 50% regardless of the sex? Okay, so it is X-linked recessive inheritance case. And now, very quickly, we'll see this question too, which says that there are some commonly used pedigree symbols and you have to choose the option which gives the correct identification for each symbol. So these are the symbols, and I hope you are aware about these symbols. So can you tell what symbols, what is the correct representation for these symbols?
Ma'am,
Which
3.
option 3? Okay. Anyone else?
Option 4, ma'am.
Option 4. Okay. So
Let's
the
see.
Ma'am,
Option
4.
4.
Actually, I thought
yeah, yeah, yeah.
2 to
Yeah, I know why the people get confused. Even I also got confused due to these numbers
Oh,
when
yeah,
they have
the
mentioned
alignment,
1, 2, 3.
alignment is different.
Yeah,
So the same thing I also did wrong while in the exams.
yeah, this
I
thing
have
happens
seen
a
the
lot.
image, but I have not seen the
All the
numbering.
numbering, right. Yeah, so it's correct will be this option D because in this, and the 2 is this one. So it is a proband male, this is a diseased female, and this is gender unknown. Yeah, it's clear. I think there's no need of explanation for this. So let's proceed to next question. So in this, the pedigree shown below represents the dash pattern of inheritance and the red arrow marks dash. So now you have to observe this pedigree and you have to see that what kind of pattern of inheritance you observe. So I
It's
hope,
dominant
okay. So it's X-linked dominant and red arrow marks an affected female. So,
carrier.
so you are saying, uh, so out of these four, which option do you think? If the female is
Option 4,
carrier,
ma'am.
X-linked recessive, a carrier
Option
male,
3, option
or
3.
option three, X-linked
X-linked
recessive.
Option
recessive and
3.
carrier female.
Okay. So the correct option is this option C, X-linked recessive and a carrier female. So it's quite clear from here only that the female is a carrier, and female is a carrier only when it is a X-linked recessive. Because if it is X-linked dominant, then the female would have shown the diseased state and not the carrier state. So carrier is only observed when there is the recessive trait, and then X-linked recessive trait is uncommonly seen in the female carriers, who usually have at least some cells with an unaffected and active X chromosome. However, female carriers can pass the trait to their sons, who will be affected because the females they are passing one of their X chromosome to their sons. So even if they are carriers, but when they pass it to their sons, so then the sons will always show that the disease. So that's why the correct answer will be the X-linked recessive and the female is the carrier female the red arrow represents. Okay, so let's move to next question. It's quite easy. So next question is a bit difficult one. So it says that Manu marries his cousin sister and gives birth to a child with cystic fibrosis, autosomal recessive disorder. Given that Manu and his wife are normal, with one of their grandparents also showing the disease, what is the probability of second child born to the couple to develop cystic fibrosis? So for this, I think it would be better if you draw it and then you observe that what would be the ratio for the second child born to develop the cystic fibrosis. So there are these options. As I don't have anything to write, so you can draw if you want. So one thing you need to focus on is that they are saying it is a autosomal recessive disorder, right? So it would be present in the autosomes. And then they are saying, then they are giving another hint that is one of their grandparents, as the Manu and his wife, they are cousins to one another. So that means their grandparent would be same, and their grandparent is showing the disease. So in both Manu and his wife, as they are normal, that means they are carrying the this recessive means they both are acting as a carrier. So if we talk about the phenotype, so you can assume that the phenotype of Manu and his wife would be if small is a recessive and capital is a dominant form, then for cystic fibrosis, small a form is carrying the cystic fibrosis, then both have capital A small a and capital A small a phenotype. Now they will be giving birth to their child, so all of their children, irrespective of their gender, the probability to develop cystic fibrosis would be 1 by 4 because they both are having the carrier genotype. You can say capital A, small a, capital A, small a. So there are 1 by 4 probability that their second child will show the cystic fibrosis. As I don't have anything to show you by writing, so is it clear that why it is 1 by 4? So you can see in the reasoning thing that as their grandparent was showing the disease and it is lying in the autosomes, so this means it is a recessive form. So in both Manu and his wife, this recessive allele would have been present. And then when they are giving, means when they are forming their own generation or own offsprings, then for each offspring they would be passing this defective allele with the probability of 1 by 4. So it's means it's clear with this that as the both of them have this capital A small a.
So it can be drawn using the Punnett score, right?
Yeah, with that also you can do. But as I
So in
don't,
case of
yeah.
ma'am, so one question regarding regarding this only. So
Hmm.
if a normal man, oh sorry, or a carrier man, if a carrier man is married to a normal female, what's the chance of their child is affected? So
So you are saying carrier male. So carrier male means you are talking about the autosomal recessive disorder in a male.
yeah,
So if autosomal recessive disorder in the male is capital A small a and female, so what about the female? Is she a carrier or she's
normal,
normal?
normal.
Yeah, so that means she has a small a. So that means what is the chance that, sorry, that female will be having a capital A, capital A. So which means that they will not, means yeah, they would never, no child would be affected, right? Because as
Carrier,
the,
carrier could be born.
yeah, child means their offspring would not be defected, but yeah,
50% will be
their
carrier.
fif- yeah, 50% would be the carrier, right? Because one is capital and one is small. Yeah, 50% of their offsprings will be carrier, right?
Yeah.
Yeah. So is it clear why this ratio is 1 by 4? Because in this case, both were having this heterozygous form, capital A small a, capital A small a. So that's why there are 1 by 4 chances that their child would have this cystic fibrosis allele.
This problem is a little bit less complex because it has already mentioned that
Yeah,
it is
recessive,
autosomal recessive.
right,
So in case if it is not mentioned, then only that it becomes a tricky question. It would be a
right.
bit difficult.
Yeah, right. Then it would be difficult to identify the genotypes of the parents. Yeah, so I don't know what are the questions in your assignment. Maybe they would be more trickier than these questions. So we will be discussing your assignments also when once that the date of submission would pass, then only we would be able to discuss the assignment questions as well. So if you have any doubts or any questions if you have in your mind, then also you can post those questions. I think these provide you some form in which you can ask the questions from the tutors
So
or
if no one comes, then I can move ahead with one more query. Actually, one more I have.
okay, okay. So say what's the query?
May I?
Yeah, sure.
Yeah. So I have little bit less knowledge uh, in how the restriction enzyme actually digests any DNA fragment. So how does it digests?
Okay, so you have doubt other than this about the restriction enzyme digestion.
Yeah, yeah.
So normally when
And
we talk about enzymes, so when we say restriction enzymes, so they have a property. So restriction enzymes do not cut anywhere. They have this property that they recognize a particular sequence. There is a recognition sequence which is present in a particular, you can say, a particular DNA or RNA. So due to which it recognizes those sequence and then produce a cut. So where they will cut it depends on the mechanism of that particular restriction enzymes. Certain enzymes they produce sticky ends. There are certain restriction enzymes which produce the blunt ends. So I hope you have some idea about the sticky and blunt ends. So, for example, if there are different recognition sequence lengths, certain restriction enzymes could recognize a sequence stretch of six uh, nucleotides or six residues long, whereas there are certain enzymes which recognizes the sequence which are four nucleotides long. So when sticky end is produced is that in that case is when the cut between that recognition sequence is not equal, when it is unequal. For example, in one strand it is cutting at the position means after one, in another strand it is cutting after position five. So in that way there will be overhangs produced. So that are sticky ends. And blunt ends are formed when the restriction enzymes are digesting exactly at the middle, that is dividing both the strands into the equal length fragments without any overhangs. So that is the blunt ends. So the restriction enzymes, the mechanism behind this is because these enzymes have some, uh, you can say there is some signal capturing form due to which it recognizes the sequence, it binds to it, and then it cleaves it. So if we talk about in the form of components, then we can say one component is for recognition and another component is for breaking. So I don't know what is your exact query. Are you asking about that or are you asking
No,
about
that
the patterns?
like suppose is there any thumb rule for the for a specific way the restriction endonuclease enzyme digests any fragment? So in case of circular or linear DNA, does it digest in the same way like it does, or is there any
Yeah,
other specific rule?
yeah. So restriction, it's already mentioned for each restriction enzyme. We have already the recognition sequence where it cuts, which type of cut would be produced. We already have the information. Even if it is a circular DNA, linear DNA, or any kind of DNA, restriction enzyme has a defined mechanism. They will identify that particular sequence, recognition sequence only. It will not bind to any other sequence. So if we talk about restriction enzymes, then they are very much means, you know, it's thumb rule that this particular enzyme would digest this particular fragment in this particular way,
So I
irrespective
came
if
across
yeah.
some formula regarding digestion. Is there any formula specific to that? I think n or n plus one, something like that.
Formula related to n and n plus I don't know about any form any such formula means with which you can calculate.
Like suppose if the question
With
being asked
yeah.
that these are the restriction enzymes and you have to tell
So
us that how many fragments could be
fragments, yeah.
generated. Yeah,
So that the formula you're talking, that is that you are talking in the terms of number of fragments that are being generated.
yeah.
Because if if you see there are two means we observe two cases, okay? Okay, sorry. So if we take two cases, one is circular and one is linear case, and in both the cases there are two sites for two enzymes to digest. Okay. So if we talk about circular one, if there are two sites where we can perform digestion, then whenever there will be digestion, then how many fragments will be formed? Only two, because it was circular. But if there was a linear fragment and then there are two restriction sites, then it would produce the fragments that would be three. So whenever a linear fragment will be digested by the same number of enzymes as the circular, then the linear one will form or will always show one extra fragment as compared to that circular form. So I think
So
that's
why
why
it's
you,
so? Why it's so? That's the confusion I have actually.
because it's not connected. In circular, you have a complete circle. When you will cut a circle, when you cut the circle from two places, then you will only get two fragments. But if you have a long thread and you cut it from two places, then you will get three fragments, right? Is it clear? Because in linear, both the ends are exposed, but in circle there is no end exposed. So if you cut it from two places, then only you can expose its ends. But in linear, both the ends were exposed. So whenever you cut it at two different sites, then there will be three fragments that would be generated. This would be more clear if you draw it and then you see it. Why is it
So
so?
is there any like this you said that
Hmm.
restriction endonuclease acts on the stick and the blunt end. So uh, is it correct that the circular DNA can only be cut with the restriction enzymes which I think targets the blunt ends?
No, actually, this is not the case. There could be blunt ends and sticky ends, and for example, the circular ones, there are major cases which says that these in the circular cases they produce the sticky ends, because if you talk about the vectors, right? So why the restriction enzymes are more studied? Because when we talk about the recombinant DNA technology, so in that we talk about the vectors which carries the external DNA. So in that, what happens is that circular plasmid is taken and the restriction enzyme digestion is performed. So in those cases, the restriction enzymes which digest that particular plasmid, they produce the sticky ends. And why the sticky ends are produced? Because then it would be very helpful, because that foreign DNA will also be cut with the restriction enzymes, with the same restriction enzymes. So both of them would show the same sticky ends, and then they would be means easier to kind of say, we can say easier to fuse both of them. So it's not the case that in the circular form only those restriction enzymes will digest which produce blunt ends, but mostly sticky ends performing restriction enzymes are used if we talk about recombinant DNA technology. So there's no variation of the enzymes if we talk about circular or the linear DNA. Restriction enzyme work is same both for the linear as well as circular.
So how the minute amount of the restriction in the nucleus are
Hmm.
being added into the, I think, Petri plate. That is how I think, I don't know exactly, but the circular DNA are being cut, then digested, then they are ligated. So how they are done under the microscope, or something else like imaging technologies. I don't know exactly how to do that.
Okay, okay. So as you have seen the question which we discussed earlier, in which we talked about Prader-Willi, and we saw that the graph in which the paternal, maternal, means father, mother, and patient bands were observed. So whenever the digestion is performed, right, so different fragment lengths are generated. So these are studied by the means if you have idea about the electrophoresis, so what happens in there is that we observe, so we believe that the fragment which are larger in length, or the fragments which are smaller in length, so on the basis of the length of the fragments, the bands would be observed at different places. So to observe that how many cuts are produced, how many fragments are generated, and what would be the length of that fragment, we observe it under by performing the electrophoresis. So means with the help of electrophoresis, the DNA moves under the charged conditions on the basis of its length. So the bands which we obtain, with which we can say that how many fragments are generated based on the number of bands that are produced, intensity of bands, because sometimes if the two bands or the two fragments are of same size, then that particular band will be more intense as compared to the another band. So to observe that, we do not observe under the microscope, but we perform this electrophoresis and everything to observe that if the digestion is accurate or what kind of digestion is performed. This is very helpful in the paternity testing as well. When we compare the child with the father's patterns or the bands and the mother bands, then we could say that means who is the father of that child. So for that also this restriction enzyme digestion is helpful. Is it clear now?
Yeah, yeah.
Yeah.
So, like you said, that the electrophoresis is being used for
Hmm.
detection at how the blends of brands are present. So,
Right.
can it be digested, or sorry, extracted out of the gel, and be used for further proceedings like cloning?
Yeah, you can extract that out, I guess, that particular segment of DNA. Okay, so you are saying that so you have obtained those fragments, and now those from those fragments, how we are going for the cloning? Is that your question?
Yeah,
That
yeah,
it's already, I mean, divided into different chunks?
yeah.
Yeah, so that from that particular gel in which that electrophoresis is occurring, polyacrylamide or agarose gel, any means depending on the type of gel we are using. So from those gels, those particular fragments are extracted, and then those are transferred to another uh, medium or another cell where they are being cloned. So in most of the cases, they're being put into the E. coli where that means those fragments are being transferred to that particular, you can say, host in which it would um, incorporate the another foreign DNA. Means those fragments are as it is entered into the host for the cloning. We do not, I means, yeah, means from those gel only we transfer those
During the
fragments. Yeah.
during the marker selection,
Hmm.
during the cloning, so we add fluorescent tags into it, right? So that we
Right.
can visualize that whether it has, I think, transformed or not. Fluorescent
Yeah,
tags.
yeah, yeah. Fluorescent tags are also added, right.
Okay.
Yeah.
And regarding the patented test, so I know exactly the time is exceeded.